Hello students. So in today's class, we will learn about transduction as the gene transfer method. As such, in bacteria, three different gene transfer methods are identified that includes transformation, transduction, and conjugation. And in today's class, we'll learn in more details about transduction as a gene transfer method. So transduction is a method of gene transfer which includes or involves bacterial viruses, which are known as bacteriophage, that will transfer the DNA from the one cell to another cell. In general, there are two different methods by which viruses can mediate gene transfer, known as generalized transduction and specialized transduction. In generalized transduction method, there is no control over which gene sequence can be transferred from first host to the second host. While in specialized transduction, only the specific regions of the first host chromosome are transferred into the second host. So let us discuss in more details about generalized transduction. So here in the first part of the image, we can see a typical lytic cycle. So during the lytic cycle, the phage will first attach on the surface of the bacterial cell and insert its DNA inside the bacterial cell. The phage DNA will then enter into lytic cycle, wherein it will take over the bacterial machinery and use it for making more copies of its own DNA. Late during the lytic cycle, the transcription and translation process will lead to synthesis of a lot of phage capsid proteins. Sometimes during the lytic cycle, the host DNA also gets degraded and it gets shredded into smaller particles, which may imitate the size of that of the viral DNA. This is why sometimes by mistake during the packaging stage, some phage particles might be produced that will contain the bacterial chromosomal DNA instead of packaging the phage DNA. Such particles or such virus particles are known as generalized transducing particles, basically because they are containing any random DNA sequence. So there is no control over which DNA sequence is entering inside the phage head here. These generalized transducing particles can now begin the infection in the second host by attaching to the surface of the bacterial cell, and then the DNA will enter inside the recipient cell. Now, once inside the recipient cell, these genes that are present on the part of the DNA are not viral genes, so it cannot undergo any lytic cycle. Rather, these are the chromosomal genes that are brought to the second host from the first host, mediated by the virus particles. So depending on sequence homology, you can have formation of hybrids by homologous recombination reaction, and that will lead to a generation of transduced recipient cells that will have new phenotypes. The next slide talks about specialized transduction. So, specialized transduction method is extremely efficient method of gene transfer, but it is selective in that it will allow only a particular sequence, the small region of the bacterial chromosome that is adjacent to the region where the phage DNA has integrated into the bacterial chromosome when the phage DNA enters into lysogenic cycle. So basically, specialized transducing particles are generated when the phage DNA will first enter into lysogenic cycle. That is, it will integrate into the host gene. Now, this integration process does not take place at any random location, but there are certain sequences within the bacterial chromosome which are known as the at sites, AT stands for attachment over here, and these are again sequences of homology between the lambda phage DNA and the bacterial chromosomal DNA, which allows integration of the phage DNA into the genome. So these are present at specific locations within the chromosome, and adjacent to these AT sequences are present specific genes which encode for utilization of sugar galactose. So basically they contain galactose utilization genes. Now, when the phage is induced, whenever it is coming out of the lysogenic stage and entering into lytic phase, during which time there is excision or disintegration of the phage genome from the chromosomal DNA. And in a normal event, the disintegration is quite precise, and you will have a virus particle, viral DNA, circular viral DNA generated that will enter into a normal lytic cycle, generating more virus particles that can infect a new host. However, in a very rare event, what can happen is that the disintegration process is such that it is imprecise. Okay, so when this incorrect or imprecise disintegration happens, then by mistake, some of the adjacent chromosomal DNA will get incorporated into the phage DNA, and some part of the phage DNA may stay back on the chromosomal DNA. Now, since all the genes that are required for the lytic cycle are present, partly they are present on the viral DNA and partly they are present on the chromosome. So the virus DNA can now enter into a lytic cycle where it is generating many copies of the viral DNA that is actually containing a small segment of the bacterial chromosome. Then packaging will happen as normal, and these virus particles that are generated after packaging are known as specialized transducing particles, because they contain, all of them will contain basically the adjacent portion of the genome that contains the genes for galactose utilization. So in this way, when these transducing particles, specialized transducing particles will infect a new host, they can transfer these specific genes for galactose utilization into the second host, that will lead to change of phenotype, that is gain of galactose utilization property. So that's it about transduction as gene transfer methods. Kindly like and share if you like the content, and subscribe to get direct updates of newly uploaded videos. Thank you.